Hello everyone, welcome from Team Nova Bio. Today we are going to discuss the basics of enzymology. In this video, points included are history of enzymes, introduction, characters of enzymes, enzyme nomenclature and classification. First, we will discuss history of enzymes. The study of enzyme called enzymology evolved with the discipline products of biochemistry itself. It started with discovery that ethanol and CO2 are the principal product of sugar decomposition by yeast. The enzyme specificity as lock and key mechanism was worked out by Emil Fischer in 1894. In 1926, J. Sumner crystallized the first enzyme urease from jacqueline, which catalyzes the hydrolysis of urea to ammonia and carbon dioxide. In 1963, the first amino acid sequence of the enzyme biovine pancreatic ribonuclease A was reported. In 1965, hen's egg white lysozyme X-ray structure was elucidated. Now we will discuss the introduction of enzyme. Enzyme is a biocatalyst. Enzyme increases the rate of biological reaction and remain unchanged in the overall process. This is a remarkable molecular device that determines pattern of chemical transformation. Enzymes have several properties that make them unique. There are nearly 2000 different known enzymes. Now we will discuss characters of enzymes. Enzymes are remarkable and highly specialized proteins with catalytic activity. Most enzymes are proteins, with the exception of small group of catalytic RNA molecules. All enzymes are proteins. They have high degree of specificity for substrate or reactants and accelerate the specific chemical reactions without formation of byproducts. They function in dilute aqueous solution under very mild condition of pH, for this neutral pH, and temperature is 30 to 40 degrees Celsius for animals and 20 to 30 degrees Celsius for plants. Enzymes are the functional units of cell metabolism. Acting in organized manner, they catalyze hundreds of stepwise reactions. Like catalysts, the enzymes do not start the chemical reaction nor change its equilibrium, but enhance the rate of reaction. Enzyme lowers the activation energy required to overcome barrier of reaction. The catalytic activities of many enzymes vary in response to concentration of substances other than their substrate. Enzymes have high molecular weight. 6000 kilodalton for bacterial ferredoxin, 25,000 for catalase, 4,82,000 for urease and 46 lakh for pyruvate dehydrogenase. Therefore, they occur in colloidal state. Enzymes are not imported. Every cell produces its own enzymes. They may be active inside cell, for this endoenzymes or intracellular enzymes, example is mitochondria, or they active outside of the cell, for example exoenzyme. Example is digestive enzyme, amylase and protease, etc. Enzymes are now used in detergents and dinner tablets are used in production of cheese. When substrate goes in transition state, the reaction will speed up. When enzyme is added, then enzyme substrate complex stabilizes substrate in transition state. Enzyme will interact with substrate in ground state, but in transition state, substrate gets fit into enzyme to lowering of activation energy. Now we will discuss nomenclature and classification of enzyme. Systematic functional classification and nomenclature of enzyme was adopted by International Union of Biochemistry. Most of enzymes have two names. The first name is the short recommended name, convenient for everyday use. For example, carboxypeptidase A. For the substrate describing enzyme catalytic action. Second is more complete systematic name, which is used when an enzyme must be identified without ambiguity. The systematic name is the name of substrate followed by a word ending in ase, ase. For example, same carboxypeptidase A has a systematic name peptidyl amino acid hydrolase. Classification of enzymes. Enzymes are classified and named according to the nature of chemical reactions they catalyze. There are 6 major classes of reactions that enzyme catalyze as well as subclasses. Now we will discuss a chart of class of enzymes, type of reaction and example of enzyme. First class is oxidoreductase, which carry out oxidation reaction. Example is lactate dehydrogenase. Second class is transferases. Type of reaction is group transfer. Example is nucleoside monophosphate kinase, MNP. Third class is hydrolases, which carry out hydrolysis reaction, means transfer of functional groups to water. Example is chymotrypsin. Fourth class of enzyme is lyases, addition or removal of group to form double bonds. Example is fumarase. Fifth class is isomerases, which carry out isomerization, means intramolecular group transfer. Example is triose phosphate isomerase. Sixth group of enzyme is ligases, which carry out ligation of two substrates at expense of ATP hydrolysis, amino acyl tRNA synthetases. This is the basics of enzymology. Thank you for watching this video. If you like this video, please subscribe for updates and knowledge. Team Nova Bio.